Okay, good morning, everybody. Good morning, Dr. Sufyan, Dr. Behara, and all students of Marine Science and Technology Department at IPB. Thank you for joining us in this general lecture. Today, we will listen the experience that Dr. Sufyan and Dr. Behara on behalf of subject the marine bio-optic and marine biodiversity. So for the first lesson, it will be Dr. Sufyan. So let me introduce myself. My name is Riza Pasaribu. I am one of the lecturer of Department of Marine Science and Technology at IPB University. And thank you for coming to this, I think, this beautiful event between our two universities, the exchange lecturer for this general lecturer. So the method of this general lecture is Dr. Sufyan will be the first, and then Dr. Behara will be the second. And during this session, all of students, you can ask, you can have the discussion, and you can ask them. You can ask Dr. Sufyan and Dr. Behara, and you in the middle of the session. So you don't need to wait me or the moderator to give you the time. So you can just raise your hand, and then maybe Dr. Sufyan or Dr. Behara will tell you if you want, if you ready to, if they're ready to give you some time to give them the question. So I think they will be all. So Dr. Sufyan, I will read your short CV first. So Dr. Sufyan Idris is a senior lecturer at the Faculty of Science and Marine Environment, University Malaysia Terengganu. He received a bachelor degree in marine science and master degree in remote sensing and GIS from the University Putra Malaysia, and his PhD in satellite oceanography from the Bangor University, UK in 2012. His research interests includes marine optics, bio-optics, ocean color remote sensing, with a specific focus on regional ocean color algorithm development and applications from satellite ocean color, and has several publications in this field. His past work includes bio-optical modeling of phytoplankton size class and size-specific primary production, remote sensing of coral reef health, coastal upwelling, and phytoplankton biomass, and impacts of river plume fronts on plastic debris. He has taught satellite oceanography, remote sensing and GIS, marine science, and ocean atmosphere and climate change at the university. So that will be all, Dr. Sufyan. The time is yours. Thank you. Dr. Sufyan, still mute the.
Okay, so Assalamualaikum warahmatullahi wabarakatuh, and a very good morning. Actually, it's already afternoon in Malaysia. Okay, it's 12 o'clock. Okay, so first I would like to thank Dr. Riza Pasaribu, okay, and from the Department Ilmu dan Teknologi Kelautan, okay, Fakultas Perikanan, and the Institute Pertanian Bogor, for inviting me or inviting us to give a small talk, okay, to your students. Okay, so I really appreciate your invitation. So actually, I had been to IPB, I think 20 years ago in 2002. So we had a summer event on the remote sensing of water quality. Okay. During that time, okay, so I think I spent nearly 2 weeks in IPB. It's a very beautiful campus in IPB. So we also went to the Pulau Seribu. Okay, I think actually 3 hours from Jakarta. Okay, so I think I would like to share my slide. Okay. I never use Google Meet and how to share my slide. Yeah. Advanced sharing. Dr. Riza, how to share
you can find the green
Okay.
share screen. Yeah. Just click on it.
Multiple participants can share this one. Okay. Yeah. Screen number 2. Okay. Because I'm using 2 screens, so the computer, the camera on the first screen or monitor, and the slide in the second screen. So I think I have to stop. Okay, for a while, I have to move my slide to screen number 1. Okay. Okay, I cannot move the cursor on the sharing button. So how to?
Actually, we already seen your share screen before.
Yeah, but I cannot share. The mouse is not moving on the share button. It's already shared or
Not yet.
not yet? Okay, screen. Okay, can you see the
Yes,
slide?
yes.
Okay.
Yes, already.
Sorry. So, okay, so here is the list of topics that will be covered in my talks. Okay, so I will start with the definition of primary productivity. So this topic is very important actually to understand the bigger picture about the productivity processes in terms of the relationship between light and photosynthesis, okay, and different types of primary productivity. So in this way, we can understand the concept of productivity and what parameters, okay, or variable that have been used to measure productivity, okay, or primary production, both in situ, okay, and from satellite observation. So after that, I will talk about the sea to space measurement of primary production, what methods are used to measure in situ, and the concept of satellite measurement. And the next 3 topics, okay, we look into the different types of primary production model, limitation of satellite measurements, okay, and lastly, a little bit about the seasonality of primary productivity, especially in the southern coast of South China Sea. So, okay, what is primary productivity? And similar to a human being, so phytoplankton in the ocean or in the lake, they need energy for their sustainability, to grow or to respire, to reproduce, or to move from one location to another location. And for that purposes, they need to produce and store energy, okay, in their body, okay, or in the ecosystem, either as a biomass or chemical energy, okay, in terms of the organic matter. So the ultimate source of energy is the sun. So maybe some organism in the deep ocean, they do not need solar radiation, because they can use the chemical energy, okay, to produce, okay, their energy. And okay, the question is how does energy, okay, especially from the sun, okay, get stored within the ecosystem, and how the energy from the sun get converted into the biomass. So the conversion of energy from solar radiation, especially, okay, start, okay, at the lowest tropic level, or known as photosynthesizers, okay, or organism, okay, that carry out photosynthesis, which are usually going to be algae, okay, and phytoplankton, plant, or bacteria. So these organisms are known as autotroph. Okay, auto mean self. Okay, troph mean nourishment, or okay, making food. Okay, and because of their ability to produce their own food, okay, in terms of the organic matter from the solar energy and inorganic source, and because of these autotroph produce their own food, okay, they are called producers, okay, or primary producers. Okay, and they form as a base, okay, in the ecosystem energy food chain. So how does the energy get stored in their body? Okay, so the energy is stored in the cell, okay, as a organic molecule that contain change of atom or change of carbon. And with this change break apart, okay, the stored energy, okay, will be released. So this carbon will break apart, okay, as, okay, when the molecule metabolize, okay, when the molecule respire, when they reproduce, or when they die and decompose. Okay, so, okay, so we have to ask, okay, how these carbon bonds. We talk about carbon, okay, how this carbon bond is coming from. So the carbon may come from the air in the form of carbon dioxide, okay, through the process of photosynthesis, okay, or, okay, dissolve carbon dioxide, okay, in the water column, okay, through the process of photosynthesis. So actually, photosynthesis is a process of carbon fixation, okay, into the biological molecule, okay, of the organism and plants, okay, especially, okay, the green plant, okay, is very important, okay, to fix the carbon, okay, from the atmosphere, or from the dissolved carbon dioxide, okay, for the outside. So as not all solar radiation can be absorbed and used to produce energy, because some will be reflected, and also because some can, and because the efficient process, okay, in converting, okay, solar energy into the organic matter, okay, is depends on the concentration of pigment. Okay, so we know that the green pigment absorb light in the blue. Okay, that's a basic concept, okay, when we learn about the ocean color. Okay, so the green pigment absorb light in the blue and the red, okay, especially at 440 nanometer, okay, and 670 nanometer, and they reflect, okay, the green wavelength. Okay, that's why we see, okay, the green, the leaf greens. So the pigment absorb only in a very narrow range of light, okay, and not all solar radiation wavelength can be used to produce energy. So on average, actually only 10% of energy get transferred, or stored, or retained as a biomass at each trophic level. So why it's only 10%? Because not all producers get eaten, and the process of consuming actually needs more energy, and also they need energy to respire, they need energy to metabolize, and they need energy, to for waste product, and especially for accretion. So we move to the concept of productivity and the definition of productivity. So actually, what is productivity? So we know that productivity, or the marine or ocean productivity, we can group into 3 types. First is a primary productivity. Number 2 is a secondary productivity, and number 3 is a community productivity. When we talk about productivity in terms of the ocean or marine productivity, so there are 3 types of productivity. So primary productivity is a productivity at the producer level. Secondary productivity at the consumer level, and community productivity, we can say that is a total productivity, that coming from a producer and a consumer. So in the broad term, productivity, we can say as the rate of biomass production, or the amount of food energy produced or obtained or stored by a particular trophic level, per unit area per time. When you talk about productivity, so it involves area and time. So for the primary productivity, it usually a rate with the fixation of carbon by the photosynthesis process. So it usually expressed in gram of carbon bound into the organic material per square meter and per day. So the unit is a gram carbon per meter square per day. Sometimes if we want to measure for months, so we can divide by months or per month or per year. So we can define the primary productivity as the production rate of energy in biomass, carbon converted into the organic molecule, through the process of photosynthesis. So there are 2 processes that get involved to calculate the primary productivity. One is a photosynthesis, another one is a respiration. So respiration is the release or the breakdown of energy, and then photosynthesis is the how the energy or the uptake of energy or the production of energy, especially by phytoplankton. And then we move to the next topic about the GPP, and NPP. So maybe all of you have heard about the GPP, gross primary productivity, and NPP, net primary productivity. So for the primary productivity, we can divide into 2, GPP and NPP. So GPP is the total of organic matter produced, that includes the energy used by producer to respire. Or we can define it as a total energy captured by the photosynthetic organism. And what is a NPP, net primary productivity? Net primary productivity is the remaining energy or biomass after accounting for energy loss due to respiration. Because photosynthesis and respiration occurs at the same time. So if you know the GPP, okay, and respiration, so we can measure, okay, NPP, okay, by subtracting respiration, okay, from GPP. So actually in the real world, okay, actually it's very difficult to directly measure GPP because photosynthesis, okay, respiration, as I said before, occurs, okay, nearly simultaneously. So respiration can occur day and night, okay. So daytime respiration is known as a light respiration, and nighttime respiration, okay, is known as the dark respiration. So, but the most dominant respiration is actually the dark respiration. Okay? So, okay, we can okay, separate or we can define clearly, okay, what is a GPP or NPP, because some ocean color product, okay, they produce a GPP, okay, and some, okay, they produce NPP. Okay? So NPP is a net primary productivity, okay, the total productivity, okay, minus, okay, the respiration energy used for respiration, okay, for the gross primary productivity, okay, is the total, okay, energy or biomass carbon uptake, okay, by the phytoplankton. So we move to the next topic about the controlling factors of the marine productivities. So, okay, there are two main factors, okay, that control the primary productivity in the ocean. The first factor is light availability, and the second factor is nutrient. But in this talk, okay, I only talk about the light because light, okay, is very related to the satellite, okay, observation. So, okay, light are the most significant factors, okay, that regulate the ocean primary productivity. And although other factors, okay, such as carbon dioxide, temperature, or overgrazing, okay, by consumers, especially by zooplankton, who also limited the primary production in the ocean. But the impacts actually are localized, okay, which especially occurred, okay, in coastal waters, okay, or in the outwelling areas, okay, or areas that receive, okay, the industrial thermal plume, for example. So light is very important to control the primary production, okay, since solar radiation cannot penetrate deeper into the ocean. So in this open ocean, the penetration of light actually is only about okay, or only until 200 meter, okay, but it's less than 20 meter in the coastal water. So in the coastal water, light penetration okay, actually depends on the wavelength of light, okay, and the amount of particle, especially suspended particles. And for example, in the turbine coastal water, the absorption of light, okay, by particle reduce the penetration light. So photosynthesis, okay, is only restricted, okay, in the upper layer. So most of the primary production occurs in the photic or euphotic zone. So I believe most of you already know about this zone. Okay, euphotic depth is a depth range, okay, where photosynthetically, okay, active radiation is about 1% of the surface value. So below this photic zone, we have a aphotic zone, okay, where PAR or light, okay, is less than 1% of the surface value. So in this zone is traditionally assumed that respiration rate, okay, is higher than the production rate. So what is a compensation depth and critical depth? Okay, these are very important because okay, from this compensation or critical depth, okay, we are able to determine, okay, the optimal photosynthetic rate. Okay, so optimal photosynthetic rate is one of the variable that used, okay, in a remote sensing model, okay, to measure primary productivity. So compensation depth actually is a depth where the rate of photosynthesis, okay, for an individual organism, okay, equals the rate of respiration, okay, by that organism. Okay, for the population, it's not as a critical depth. Okay? So although it's a similar term, okay, but okay, the concept, okay, one is applied for individual, okay, it's not as a compensation debt. For the community or population, it's not as critical debt, okay, meaning that, okay, at which the photosynthesis rate equal the respiration rates. So this debt actually located at the euphotic depth. So for below this depth, okay, there is a net respiration, okay, net respiration, okay, where the respiration rate is higher than the production rate.
Dr.
So for
Sufyan,
some, yep.
sorry to interrupt you. The picture is too small. I think you should.
Small.
Yeah, I think you should right click on that your presentation, right click, and then hide presenter view.
Okay, this one is okay.
Yeah, that one is okay. And then
because I use two screen actually, two monitors. Okay, when you you
please
do
back, please back to your screen before.
Okay. I think no need to view in the presentation slide. I think it's a.
Yeah, this one. Can you full screen that? I think. Yeah.
So okay. So it's jump to the second monitor.
Yeah. Okay. The second monitor, and it's okay. You can use your second monitor, and then you just right click and hide presenter view. Going back again.
Going back.
I think it's better this one because the picture is bigger.
Okay. Sorry.
Okay.
There's a problem when using two monitors.
Yeah, I think it's okay if you like this. Okay.
Okay, so these are okay. So I will continue. Right? So for some species, okay, especially below this euphotic depth, okay, they will form cysts or resting spores, okay, to reduce their metabolic rate and to maintain their biomass or energy. Okay, so we can see that the productivity is minimal at the surface, okay, because of too much sunlight and low nutrients. And too much sunlight actually could saturate the photosynthetic cell and inhibiting photosynthesis. So the productivity is actually greatest at the subsurface layer, especially below the thermocline layer, because there are abundance of nutrients, okay, and the light and intensity is not too high. So the primary production is actually okay, determined as the integral production, okay, from the surface, okay, until the compensation depth, okay, or until the euphotic depth. Okay, so it's not okay measured, okay, at the surface, but okay, actually we take, okay, the whole productivity, okay, primary productivity or production, okay, from the surface until the euphotic depth. Okay. And then, okay, if you look at the photosynthesis rate, again, the light intensity, okay, this is most important when you measure, okay, primary production, okay, from the satellite, okay, we have to know, okay, how to determine the optimal photosynthetic rate, okay. So if you look at the photosynthetic rate, okay, against light intensity, photosynthesis rate, okay, increase, okay, from the low light to bright light, and reaches its minimum actually when the photosynthetic cells, okay, reaches its minimum, okay, and it start to saturated, okay. Okay, when okay, there is an increase in light or light radiant, okay, meaning that it cannot increase its photosynthesis rate and starts to reduce, okay, its photosynthetic capacity when you increase, okay, the light irradiance. So the reduction of photosynthetic rate is known as a photoinhibition. Okay, as a result of the excess photon or light, okay, they are not dissipated, okay, by the photosynthetic carbon. So the maximum photosynthetic capacity is known as the optimal rate of photosynthesis, or PB, okay, max. Okay, so we can see that the photosynthesis occurs, okay, below the compensation rates or points, okay, where the percentage of light is below 1% of the surface value. So the net photosynthesis, okay, can be considered, okay, from the compensation point to the optimal, okay, rate of photosynthesis. So the efficiency of the photosynthesis rate or process can be estimated, okay, if we know the photosynthetic quantum efficiency, okay, that is determined by the initial slope, okay, of this PI curve. So normally, smaller cell size such as phytoplankton, they have a higher photosynthetic efficiency, okay, than larger cell size such as microplankton. And then the determination of optimal rate of photosynthesis is very important to calculate, okay, primary production, since the photosynthesis is the only gateway for carbon, okay, in the gas form, okay, to fix into the solid form or carbon bonds and stored as energy. So there are different equations used to calculate the optimal rate, okay, of photosynthesis for determining the primary production. Okay, this is an example of a model by Platt, okay, in all 1980. And how is in situ primary production measured? So in this slide, we will look into the in situ measurement of primary production. Okay, there are different methods, okay, used to measure primary production. And the traditional one is the oxygen technique, okay, using the light and dark method. And if you make a measurement in the field, you can arrange the bottle according to the depth until the base of your depth, okay, something like this figure. And incubated the sample, for example, for 1 day, okay, and measured the dissolved, okay, oxygen at the beginning and at the end of the incubation. So you want to measure, okay, your DO or your sample in the lab, you can sample the water column at the discrete depth, okay, under different light condition, and incubated in the lab using the same light condition. Okay. So, for example, you have 2 bottles of water sample collected at similar location. One is exposed, okay, directly to light. Okay, another one is wrapped with opaque cover. Okay, so you can use aluminum foil to block out light, so there is no light penetrating the bottle. So in the transparent bottle, actually the photosynthesis and respiration, okay, or known as a light respiration, will produce and use up oxygen. So if you want to measure the production, for example, for 1 day, so we measure the amount of dissolved oxygen, okay, in the sample, okay, at the beginning of observation, okay, using the DO meter. Okay, for example, for the initial DO, we get 10, okay, milligram per liter of ppm. So after 1 day, for example, you measure the DO again, okay, in the dark bottle, for example, you get 6 ppm. So now the initial DO is 10. After 1 day, okay, only 6 ppm left. So we know that 4 ppm, okay, of oxygen has been taken up for a respiration process, okay, for 1 day. So in this way, so we can calculate the GPP. So GPP is now 10 plus 4 ppm. Okay, so you get GPP, okay, of about 14 ppm. Okay, so you can also measure, okay, the other products, okay, or productive components, okay, NPP, okay, using this method. Okay. And another techniques, this one is okay, new techniques or advanced technique, okay, using the carbon-14, okay, techniques. Okay, so okay, we use carbon-14 as a tracer, in the uptake, okay, of a dissolved organic carbon by phytoplankton. And actually, this method is very sensitive, okay, sensitive. So it's usually used in the low productive area in the oligotropic region, okay, such as in the South China Sea, okay. So area with a low productivity, okay, because it's very sensitive, okay, to the measurements. And similar to the oxygen tanning, the sample containing phytoplankton and carbon-14 need to be incubated, okay, under different light condition, okay, for at least 1 to 6 hours. And this method is based on the assumption that the biological uptake, okay, of carbon-14 label dissolved organic carbon is similar to the biological uptake in carbon-12 dissolved organic carbon. And to determine the uptake, so we have to know, okay, the natural dissolved organic carbon in the water sample, the amount of carbon-14 dissolved organic carbon, okay, added, okay, to the sample, and the amount of carbon-14, okay, retained, okay, in the sample. So actually, this technique, okay, only measure the net carbon flux, okay, obtained or stored, okay, in the primary producers. It cannot measure, okay, the respiration, such as in the bottle techniques, okay, or the oxygen techniques. Okay. So okay, so how is primary production measured from space? Okay, so we know that okay, for the primary production, it involve the ocean color okay, remote sensing. When you talk about the ocean color, okay, it's not about the color of the ocean. Okay, so okay, we can define ocean color, meaning that okay, the visible wavelength or the water leaving radiant in the visible, okay, that leave, okay, the sea surface, okay, and reach the sensor, okay, and give the color to the ocean. Okay, so we talk about ocean color, it's not about the color of the ocean, okay, it's about the water leaving radiance, okay, especially in the visible, okay, from 400 to 700, okay, that leave the sea surface, okay, and reach the sensor. So measurement of this okay, primary production, okay, is based on the visible EM 400 to 700. Okay, so the wavelength that our eyes can see. Okay, our eyes can only see, okay, 400 to 700 from blue to red. Okay, and satellite will measure the emergent flux of water leaving radiant, or known as ocean color. Okay, ocean color. And this outward flux, okay, contain information on the phytoplankton, okay, in the water column. Actually, in this process, okay, there are two important processes that happen in the water column. Okay, one is a scattering process, and another one is a absorption. Okay, so particle in the water column, okay, especially okay, organic matter, phytoplankton, and detritus, okay, they scatter and absorb, okay, light, okay, that penetrate into the water column. Okay, some will be absorbed, some will be scattered. And for the remote sensing, and especially for the ocean color, okay, what okay, the most interesting, or what we are more interested is the scattering, okay, light. Okay, it's not the absorption, but the scattering light, okay, that leave the ocean surface and reach, okay, the satellite sensor. And okay, remote sensing reflectant or water leaving radiant, okay, can also be linked, okay, into the phytoplankton threads. Okay, so from the this assumption, okay, back scattering, okay, divide by A equal, okay, remote sensing reflectant. So we know that water leaving radiant or remote sensing reflectant, okay, equal back scattering by the particles or phytoplanktons, okay, divide by, okay, the absorption properties of these phytoplankton or other particles. So actually absorption and scattering is a contribution of many particles, or contribution or additional contribution, by other particles such as phytoplankton, such as detritus. Detritus is a non-pigmented particles, and colored dissolved organic matter or chromophorics, organic matter. And backscattering actually is an additional contribution, or contribution from different particle, such as from the phytoplankton, mineral particle, and also from the water itself. So actually remote sensing reflectant, we can relate with the amount of phytoplankton with the types of phytoplanktons in the water column. So actually the magnitude and shape of remote sensing can depend on the amount and types of particle in the water column. This is an example of a study in the Malaysian waters. So we collect data for nearly 11 years to study the types of water that based on reflectant, that based on particles in the water column. So we can see, or we found that there are 5 water types, 5 water types in the Malaysian waters, and these water types are located at different regions, and can only be found in a specific season. For example, for the Strait of Malacca, most of ocean, or most of the sample is come from the class 5. Class 5 meaning that, that it has a very low reflectant in the blue and high reflectant in the green. So low reflectant in the blue because of very strong absorption by the phytoplankton especially. And then because of very high absorption, so it will create a very low reflectant in the blue. So we can see that there are other types of water reflectants. For example, in the coastal water of South China Sea, so we can get a class 3. Class 3 meaning that high reflectant in the green and also in the blue wavelength. So this magnitude, different magnitude and shape of reflectant, it can show different particles, different types of particles in the water column. And this one example of optical classification of water types, in the Malaysian waters that can be used, to identify or to measure chlorophyll concentration, especially because chlorophyll is a proxy to primary production. So in this study, there are at least 5 class with different concentration of particles. So if we can see for class 1, class 1 contain less particle, and this particle concentration increase with class number, until class 5. So this result can be used to identify or to measure primary production, especially chlorophyll pigment, from the satellite data. So RS is also affected by the concentration of chlorophyll in the water column, not only from the other particles. So we can see that high absorption, that means low reflectant, low absorption by chlorophyll, or low concentration by chlorophyll, meaning that high reflectant, especially in the blue. So how is primary production measured from space? So we know that most of the satellite data are designed to measure specific pigment peaks of phytoplankton, spectra. So if we can see the blue lines that represent phytoplankton absorption, this one is measured in the lab. So you can use a spectral paper, okay, and spectrophotometer to measure the absorption of phytoplankton. So we can see that phytoplankton, okay, absorb more light, okay, in the blue and red, okay, okay, at 443 and 670 nanometer. And most of the satellite sensor, okay, have this band, okay, to measure, okay, the specific pigment peaks of phytoplankton spectra, and these can be used to identify, okay, chlorophyll concentration, okay, in the from the remote sensing observation. So this one example of this okay, chlorophyll algorithm. Okay, so you use the blue to green band ratio. Okay, especially usually actually it's very easy to measure or to detect a phytoplankton or chlorophyll, okay, from the satellite, okay, in the offshore waters. Okay? But for the coastal waters, and because of the competition of photons, okay, between particles, okay, other than chlorophyll, so okay, it's very difficult, okay, to get the accurate result of phytoplanktons. So satellite remote sensing of primary production. So we have different types of ocean color sensors. Okay, the past sensors and the future sensors. Okay, and then the current sensor. So right now we have a Seawave, MODIS, okay, MARIS, okay, until Sentinel. So you can use that data, okay, to derive primary production. And the future hyperspectral ocean color is a PACE, okay, that okay, PACE is a hyperspectral ocean color. Hyperspectral, that means it has a very narrow range of band, so it can be used to measure, okay, different types of phytoplanktons. It's not only chlorophyll, but you can also measure cell size phytoplankton, microplankton, picoplankton, nanoplankton, okay, using this hyperspectral ocean color sensor. So it is expected to launch in 2023. Okay. And okay, for the satellite product of primary production, okay, so actually all satellite data, okay, especially for oceanography, is free. Okay, you can get it free. Okay, no need to pay. Okay, so for example, for the primary production, you can get, okay, the primary production products, okay, from the site Ocean Productivity from the Oregon State University. So it gives you different products, okay, that based on different models and different sensors used. Okay, this one example of sensor-based primary production from the MODIS, Seawave, and VIRS. Okay. And then this one is a model-based primary production. So okay, there are 3 or 4 types of model: VPGM, okay, vertically, VGPM. Okay, it's not VP, VGPM. Vertically generalized, okay, production model, and then we have a IPB model, okay, carbon-based production model, okay, CAFE, carbon absorption, fluorescent, and euphoric depth-based model. Okay. So actually, I don't know which one is accurate. Okay, so it depends on the regions. Okay, in some region maybe VGPM, okay, is more accurate. In some region maybe CBPM is more accurate. Okay. So satellite, this one is okay, for the ocean color. Okay, it's not offered any primary production product, but only the chlorophyll, okay, PAR, and so on. Okay, so if you know how to calculate the primary production, so you can get, okay, the data, okay, especially for PAR, chlorophyll, ZEU, okay, from the this ocean color product. Okay, it's also free. You can get it free. And then for the biooptical model of primary production, so okay, it involves a very complex computation. Okay, so we started from the surface irradiant or reflectant, okay, until the irradiance at a depth, at euphotic depth, and then until we measure the primary production at depth integrated. That means from the surface until the euphotic depth. So it involves chlorophyll model, it involves the photosynthetic photolite model, KD, PAR, and so on. So this one example, I will not discuss in detail about the model, but this one example of how to calculate the primary production from the satellite data. So in this model, so we have to have the PAR data, euphotic depth data, chlorophyll data, and also SST data. And then all these parameters we feed into the equation to get the primary production products. And then for the APLI, the difference between APLI and VGPM is in terms of the calculation of the optical rate of photosynthesis. For the VGPM, it uses the polynomial models. But for the APLI, it uses the you can see the power function model. That's the difference between these two models. So the for the carbon-based production model, so it involves more parameters. It's more complex computation. It involves the carbon, identification of carbon that based on the growth rate of phytoplankton. It involves the light attenuation coefficient, MLD. You have to know the mixed layer depth, and the median of MLD light, and also it involves the absorption by phytoplankton, absorption by backscattering, and so on. And lastly, the new one is a CAFE. CAFE is a carbon absorption, fluorescent, and euphotic based model. So it's a more difficult, more complex than the CB or carbon-based production model. And then how good is the satellite derived primary production? So it's very difficult to answer that question because what I said before, it depends on the region. In some region, maybe you can get a very accurate result. In some regions, you cannot get a better result. Because of what? Because of different water types, because of different phytoplankton species and type, and so on. So this one example from the study of Shi 2001 at the seed station. It's a fixed station in the South China Sea. So from their study, they found that the primary production when compared to the in situ data, it shows the overestimation of the satellite data products. So when they analyze the parameters that maybe impact the accuracy, so for the SST and for the optimal rate of photosynthetic parameter, so there are a good estimation of both product because SST is very easy or very accurate measurement from the satellite data. And for the PAR data, there is some random bias. You can see some random bias here. And for the PAR, they get a good estimator. And for the diffuse attenuation coefficient or KD, most of the satellite data is underestimated the in situ value. And for the ZEU, euphotic depth model is the satellite data overestimated the in situ data. That means the model used the euphotic depth is not accurate enough. And then lastly is a chlorophyll. We can see that there is a random bias of chlorophyll, especially at a larger concentration of chlorophyll. So in this model, we can see that ZEU or euphotic depth and KD, okay, had a more pronounced impacts on the satellite derived primary production. If you want to get more accurate result, maybe the model of ZEU and KD need to be improved, and also for PAR and chlorophyll models. And then this one example, okay, the study case in the Malaysian waters. Okay, there is a difference between global model and regional models. When you use a global model, actually it's not work. Okay, in some areas, okay, some waters, okay, especially in the Malaysian waters, we can see that for the chlorophyll concentration, there is a deflection, or there is a overestimation of chlorophyll when chlorophyll is higher than 0.5 milligram, okay, per 0.5 okay, milligram per meter cube. And similarly for the SDDs, actually that can be used to measure ZEU. Okay, so when we use the global model, okay, it shows high errors. But okay, when you use the regional model, okay, specific for that area, we can reduce that errors. And then okay, lastly is the seasonality of NPP. So okay, especially in the southern South China Sea, so we can see that high and dynamics, okay, primary productivity or production in the coastal area, but low and less dynamic production, okay, in the offshore waters. Okay, so this one is depend, okay, on the season. Okay, actually is high, okay, during the monsoon season or northeast monsoon season, okay, especially in January, okay, but low, okay, during May. May is during the inter monsoon season, okay, in the South China Sea. So we can see that the production, okay, varies with season, okay, due to the variation in chlorophyll or changes in phytoplankton biomass and types, okay, due to the light intensity between the northeast monsoon and southwest monsoon, okay, due to the water temperature during the cooling and warming period, okay, winter and summer monsoon season, and it can be affected by nutrient availability and also by the water column mixing. Okay. So this one is a monthly average distribution, for example, okay, in terms of the phytoplankton size type. Okay, this one, okay, this study has been conducted in 2019, 2017, 2019. We can see that there is changes in the phytoplankton cell size, okay, between season. So we can see that, okay, during the southwest monsoon or summer season, okay, phytoplankton actually dominates, okay, the study area, especially in the South China Sea. But okay, during the northeast monsoon, when okay, the nutrients is high and temperature is low, okay, large cell size such as nanoplankton and microplankton dominates, okay, in some area. Okay, so larger phytoplankton cell size has had higher contribution, okay, than the smaller phytoplankton to the carbon uptake rate, okay, of primary production. So phytoplankton types, okay, is also, okay, have a, okay, contribution to the different or the seasonal primary production, okay, in southern South China Sea. And this one is a long-term trend of NPP in the southern South China Sea. There is no, okay, constant or increasing or decreasing trend, okay, for 20 years. So we can see that, okay, there is up and down. Okay, the red color is a positive, that's been above the average for 20 years, and okay, the blue colors is below average. Okay, there is no, okay, actually it's a very constant. There is no increase or no decrease in primary production, okay, for 20 years. So this one is factors including the NPP. One is a climate variability of El Nino, La Nina. Okay, so actually El Nino and La Nina, okay, it affected the primary production, okay, for the 3-month or lag, for the 3+ month lag. Okay, so we can see that, okay, during the high primary productivity in 2011, okay, during that time is a La Nina. In 2015 and 2019, okay, and so it will produce, okay, low primary productivity during this time. Okay, so we can see that, okay, El Nino, okay, there is a lag of 3 months of the El Nino, okay, impact on primary production. And also, okay, this one is a spatial correlation, okay, between SST. SST also can affect the primary production. It's a very high correlation. It's about 0.7, okay, especially in the southern South China Sea. And also wind also can impact the primary production. And as a summary, so okay, I can say that, okay, ocean is very crucial. It's a very important, okay, to mankind and the climate system. So actually, there is a couple process, okay, between ocean and climate. When you change the ocean, you change the climate. When you change the climate, we change the ocean. Okay, so observation from space allow us to measure a range of environmental parameter, okay, in the synoptic and repeated way, like no other technique does. Okay, so it will give you, okay, many advantages, okay, over the traditional method. And satellite data provide an enormous amount of ocean observation, okay, in a range of application, okay, at the coastal area, at the local scale, regional scale, or global scales. And the complexity of the techniques, okay, for the remote sensing of the ocean need specially trained scientists, okay, with more or with oceanographic knowledge, especially, okay, from the marine science student, okay, and signal processing and statistic skill to exploit, okay, the information in ocean remote sensing data, in ocean remotely sensed data. So we want to measure, to observe the ocean, okay, what you need is a background in marine science. Okay, so those scientists are you, the marine science student. Okay? So I think that's all for me. Okay, and thank you for listening. Okay, so thank you.
Thank you, Dr. Sufyan. It's very interesting what you share to us. We still have time. So students, do you have any questions for this subject? You can use Bahasa or in English. If you use Bahasa, I will translate it for you.
Yeah, it's okay. If you use Bahasa, I will answer in Bahasa English.
Okay.
No problem. I can speak Bahasa.
Okay. Is there any? 1 or 2 question maybe? Okay, maybe I want to ask 1 question for you, Dr. Sufyan.
Okay.
Before our students, maybe they're still shy. Is the spatial distribution of primary productivity in Malaysian waters the same? It is the same every time, or is there is some, or there is a unique pattern that occurs? If so, what causes the pattern?
Actually, in Malaysian waters, I think we share the Indonesia similar. We share the same waters. The most important factors that determine the spatial or temporal distribution of ocean color product actually is a monsoon season. So we get a high concentration or high values of, for example, chlorophyll or primary production during the northeast monsoon, and low production during the inter-monsoon season. Okay, actually we can see there is a 2 things here. One is okay, during a northeast monsoon, okay, northeast monsoon from November to March. Okay, so there is a peak in concentration in January, and another one is a southwest monsoon from June to September. There is a high concentration, okay, especially in August. Okay? And because of this monsoon, so it will affect the temporal distribution of primary production, for example. And especially during the inter-monsoon, in April and May, because of the solar heating during that time. So it also impact the distribution of primary production or ocean color in this region. So I think we Malaysia and Indonesia have a similar weather pattern or climate pattern, and I think monsoon play the most significant role.
Yes. Okay, thank you, Dr. Sufyan. So, is there any from the students? Okay, Muhammad Soni Sastra Antara. Okay, your time is yours.
I want to ask a question, but first, I'm sorry that my camera cannot display my face.
Okay.
So the question is about the phytoplankton aerosol cloud ocean ecosystem, Mr. Sufyan. Could you explain more about that data? Because it's kind of new for me and I've never used this kind of data.
Okay, so thank you for your question. So I will show the slide about the PACE product. Actually, PACE is where's the PACE product? PACE is a new satellite that we launched. Actually, they planned for this year, but because of the pandemic, and they postponed to spring next year. And for the PACE product, actually is a hyperspectral ocean color sensor. This one is still in the pipe. It's not launched. It will launch in 2023, but I think right now they start to offer or invite the user to get involved or to have a tutorial about how to use the PACE. If you go to the PACE website, you can see you can see a lot of opportunities to get involved with their project actually. So phytoplankton aerosol cloud and ocean ecosystem is a NASA project. And I think this mission actually has been developed to replace the ocean color mission or ocean color website that we have. Currently, we have an ocean color website. That's the intention of PACE. So everything you want to know about the aerosol, about the dissolved organic, phytoplankton, pigment, functional type or functional groups. That means they can or you can identify the cell size group, like microplankton, picoplankton, nanoplankton, diatom, and so on, using this application because of the hyperspectral resolution of this sensor. So you can go to the this website, paceoceanscience.org.
Yes, it's a very limited knowledge about the hyperspectral.
Actually, they offer or they invite you to get involved into their activities. Or maybe you want to propose your own project. You can propose in that website.
Yes. Okay, thank you. The last one is from Farrell. It's in the chat. Is it possible to measure the value of primary productivity below the compensation depth? Because as long as the optical depth value has not reached zero, it can still be said that there is light remaining.
Okay, when we talk about the compensation depth, traditionally we assume that the depth at 1 meter, 1% of light remains. 1% of light remains. Actually, that one is a compensation depth. So for most of the model, we measure the primary production until the euphotic depth or 1%, okay, of light, okay, 1% of light. Below that level, okay, or that point, okay, there is a photosynthesis process occur, okay, on that depth, okay, but okay, because of our definition of compensation depth is a 1%, okay, so traditionally, okay, 1%, that means below 1%, okay, photosynthesis equal the respiration process, okay, that means okay, net primary production is a 0, okay. So actually, okay, when you go, okay, or you measure in situ, okay, sometimes you can get, okay, primary production, okay, is not 0 under the, okay, euphoric depth, okay, because we use it, okay, traditionally, okay, when we talk about compensation depth or euphoric depth is a 1% of light, okay, remaining. But actually, okay, the photosynthesis, okay, is higher than respiration, okay, below that depth. Okay. I hope I answer your question.
Yes. Okay. I think that will be all. Thank you, Dr. Sufyan, for sharing the knowledge to us. On behalf of our person in charge, Dr. Bisman, we would like to say thank you for your lecture in this good day. So I think we will be moved to the next session, and I will invite Dr. Begin as the moderator. Thank you once again, Dr. Sufyan, for your time. See you.
You are welcome.
Dr. Begin? Okay.
Okay.
Time
Thank
is
you.
yours. Thank you.
Pak Riza, good afternoon, students, and good afternoon, Professor Behara Satyana Narayana. I hope I
You can call me Satya. Simply.
Setia, Profesor Setia. Thank you. Good. Selamat siang ya, Prof. Setia, and welcome to the General Lecture 2020. Thank you very much for your time, yeah, for sharing with the students in Marine Science and Technology, IPB University. So before we start, I will quick introduce, yeah, about Professor Setia. So Professor, the full name, ya, Behara Setyana Raya. Nah, Setyana Rayana. Sorry. Professor Setia worked as associate professor in Institute of Oceanography and Environment and higher institution Center of Excellence in Marine Sciences in University Malaysia Terengganu. So Professor Setia originally from India. So for the education, PhD, graduated from Andhra University, India, and in subject of ecobiology and remote sensing based study of Coringa mangrove in the Godavari Delta, east coast of India. And for the master course, also in India, Acharya Nagarjuna University, in subject in zoology of aquaculture specialization. And for the undergraduate, BSc is a study about fisheries, zoology, and chemistry in Andhra University. So, and for the professional experience from 2013 until now is as an associate professor in INOS, UMT, and postdoctoral researcher in Laboratory of System Ecology and Resources Management in University Libre Brussels from 2010 until 2012, and 2006 until 2009 a lecturer in INOS, and before that 2004 until 2006 a postdoctoral researcher in key laboratory of tropical marine environmental dynamic in South China Sea Institute of Oceanology in Guangzhou, China. And there is a quite a lot activity still from after finish the graduates or undergraduates already doing like research assistant, senior research fellow in several marine laboratory in India, and mostly working on mangrove, Prof. And today Professor Setia will present about the marine biodiversity and about the development of research. Okay, Professor Setia, time is yours, please.
Thank you, Professor Beginner. Thank you so much for your kind words and encouragement. It's my pleasure to be part of this program today, and thanks to IPB for inviting me to be part of this lecture series. Yes, I would like to thank the authorities from UMT, Dr. Wong, for giving me this opportunity, and also the other people who have been supportive for this talk. I'm going to share my screen to deliver my talk. Actually, it is my talk is not entirely different from the previous talk, because I'm also going to touch upon some application of remote sensing. When I look into the agenda of the invitation, I noticed the first talk is going to be on the remote sensing. So good, I can keep the same trend goes on for the second talk with some applications on the mangroves. That means you already saw the application of remote sensing from the coastal waters or open waters to see the primary productivity. Now we can see how best we can use the remote sensing technology to study the mangroves as part of my talk. I'm representing my mangrove research unit situated in the Institute of Oceanography and Environment, University of Malaysia, Terengganu. Since my expertise are mangroves, today I'm going to talk on mangrove research and some advancements, technological advancements for effective conservation and management. It is quite useful to the people out there, definitely, because of Indonesia is hosting the world's largest mangrove cover. So it is you all are fortunate to have such a big treasure with you. So it is our customary to protect and keep the goods and services to reach for our future generations. So the present research was taken as part of my team from Mangrove Research Unit in Malaysia, and also with some other partners from Belgium, Australia, and UK, and also France. So this is a combined research. Mostly, you must be knowing there is a program called Tropimundo, Tropical Biodiversity and Ecosystems. So if any one of you are bachelor degree students, there will be Tropimundo scholarships every year. You have to apply online, and if you are lucky, your scores are good, you are lucky to join this kind of Tropimundo programs. That's where the internationalization could be improved, and that could help you to flourish in your passion of research. One thing I can tell you that I'm not going to teach you any equations in this in my lecture, so please relax. I just want to show you some applications and how best you can use some mobile applications also for your research in mangroves. So, as you all know, there are recent estimates already available from Food and Agriculture Organization in 2020. So they already come up with the area statistics of mangrove cover in globally in the world. So it is evident before 2020s we have been thinking that the mangrove cover is almost like over 15 million hectares. But now we must realize that the forest is still decreasing, and now it reached to 14.7 million hectares. Although the rate of decline is not as fast as before, but it is still decreasing. It's still decreasing. When you compare the statistics between 1990 to 2020, so it is the decline is evident, which you can see from the bottom line. That used to be 15.7 million hectares, now 14.7 million hectares. Although the rate of decline is decreased to a maximum of 2% in recently. But one thing you can notice what happened to Asia, why it is declining still at a very high, exceptionally higher rate when you compare it with the other geographical continents. So this is a really worrying situation, especially for the people, mangrove researchers in Asia, because Asia is the place that known to have lot of mangrove replantation works, isn't it? So there are so much funding available from the World Bank and so forth. So we have used after this 2004 Indian Ocean tsunami, which has been a bad incident for all of us, which we don't want to recollect. About the victims and the property and people, loss of people and the property, but still the protecting agents like mangroves on the coast are still decreasing at a higher rate. What could be the reason? This is really a question that everyone should be thinking about it. So yeah, we immediately blame anthropogenic activities. Of course, it is true. Anthropogenic interventions might be causing to a greater decline of mangroves, but probably that is not only the cause. There must be something else. For example, when I look into some kind of mangrove areas that known for historical importance, one of the area in India I'm showing to you now with a very good mangrove cover of more than 200 square kilometers. So when you compare the decadally, the mangrove loss, the current mangrove loss is more than 1 kilometer that lost into the sea. It is not necessarily to the anthropogenic intervention. It is also because of the climate change or sea level rise. So that is probably for the common people, the forest that has been lost into the sea is invisible. So it is not visible clearly. So probably people are thinking only the patches which are available. So probably somehow I began to feel that the loss of mangroves to sea erosion might have been understudied, undervalued. Probably that is also one of the crucial points which we all must be thinking about. And we have to see how much we lost already to the sea, the mangroves lost to the sea, and what are the possible remedies, actions, remedial measures we could implement. That is the time for us to think about this coastal erosion along with the anthropogenic intervention. So as you all can see, the benefit of satellite data here. So you can obtain the satellite data over a period of time at decadal intervals or any intervals which you may choose, and see the difference in the habitat loss or gain. In some areas, you might be able to have some new mangroves also. That's what we call it as gain. But at the same time, you might have areas that lost into the sea. So that's loss of mangroves. So it is, satellite data is extremely useful, because when you wanted to do such kind of survey manually, which is not possible, but satellite can give you the most authentic information over the decades. So it is once again highlight the importance of satellite data. Yes, mangroves. Yes, mangroves have been protecting agents. They are the green belts and coastal protectors. They are really good to mitigate the impact of the waves. What if we don't have any mangroves? This is what you have been seeing here in December 2021 during the cyclone. The encroachment of seawater, tides, and waves very close to the almost to the road or habitation grounds. That is really a fearful situation. So when you look at the place that you played 1 year before with your kids, and now you see the encroachment of the seawater in that particular area, it is really frightening, isn't it? It is really frightening. But mangroves also have a limitation. So not necessarily always the mangroves. Mangroves, sometimes they sacrifice, they are uprooted when you have these kind of things. That's why we began to promote, along with the mangroves, also some other coastal vegetations to be taken care of along the beaches to have a better protection to the people and also to the property. So as you all can see in this slide, the IPCC has already projected the sea level rise. For example, what I'm showing here is for peninsular Malaysia. So one thing you can see clearly, East Coast has a projection of sea level rise by 2100, about 0.6 to 0.68 meters of height in the sea level rise, whereas the West Coast is 0.5 to 0.6. So when you compare with the East Coast and West Coast, there is almost like 0.1 meter variation of sea level rise incidents. So one thing I would say, probably the West Coast being located on the sheltered environment, sheltered by the Indonesia one way, and also the maximum cover of mangroves that existing along the west coast of peninsular Malaysia when compared to the east coast of peninsular Malaysia, which is exposed to South China Sea completely. So that could be the reason that we have a bit less the coastal erosion or sea level rise along the west coast when compared to the east coast of peninsular Malaysia. This is what I was talking to you. So sometimes the mangroves are efficient protectors, land builders, but sometimes mangroves alone cannot do it. They will sacrifice and we may not be able to fulfill the task very well. So we also have to look into the sand dunes and some planted trees and also the natural vegetation along the coast like casuarina and so forth. These all things have to be taken care to have a better protection to the people and the property inside. So we cannot always count on mangroves, because mangroves cannot grow everywhere like other trees. So they have a specific areas of supporting areas. So we have to take care of all other coastal green belts or whatsoever you name it. So to have a better protection to our coast. So COP26, the conference held in Scotland, also highlighted that the forests are still decreasing everywhere, and it is time for us to really put an halt for the coastal or also the rainforest to stop the degradation at a faster declining. Especially in the case of mangrove, we always refer to the Sustainable Development Goals. There are 17 Sustainable Development Goals, and the mangroves indeed contributing either big or small to each and every Sustainable Development Goal. So see how important the mangrove ecosystem to this earth, to this planet, especially climate action, with the life below the water and so forth. So they have a major role playing invisibly. So we already have to we have realized we already realized the importance of mangrove ecosystems. The only thing, how efficient they can be conserved through different technologies like application of remote sensing for a clear cut identification of the mangrove areas, gain areas, loss areas, where exactly the impact of anthropogenic intervention taking place, where exactly the coastal is eroding. So that kind of identification of the hotspot is very important for the future. So my talk also touches upon the new technologies like drones, how you can make use of them to study the mangroves. So as I told you, the mangrove destruction is almost like a turning tide. That means not like before, it is greatly reduced, but still going on 2 to 0.4% year. We have to focus on where exactly these crucial hotspots are still available and how we can take care of those kind of hotspots to reduce to protect them and reduce the anthropogenic interventions. As a scientist or as a biologist or as a mangrove marine researcher or a mangrove ecologist, so we have to realize the fact that public perceptions, they are really extremely important to bring a change. So it is the public perceptions can easily bring a change and can effectively bring a conservation and management measures into the public society. So that's why some people still have some negative perceptions. For example, mangroves. Yes, they think, oh, mangroves are dirty wastelands. So that kind of perceptions, it is time for us to remove some of the negative perceptions. One of the things I tell you clearly, the mangroves, as you all know, they can trap the sediment. That's why we call them as land building agents. So they can build the land by trapping the sediments. But unfortunately, mangroves not only trap the sediment, but also they trap debris, which you are releasing into the mangroves. For example, you're releasing a lot of plastic, so much marine litter you have been releasing into the, so after eating food, the doggy bag is just ending up in the water. So the fishermen or nearby adjacent villages, they're releasing a lot of pollutions and then means plastic into the. So unfortunately, the mangrove root system is not is trapping together with the sediment also the plastic. So people by looking at the mangroves, you how bad you feel, like, oh, they're really dirty, they're really, but you are the one making them dirty, isn't it? So we have to, for example, mangroves, we think also, I would like to give you an example, like mangroves, people think, oh, they are the mosquito grounds. But indeed, we are the one leaving the plastic into the mangroves, and some of the bigger plastic, like the plastic trays, they trap the fresh water, the rainwater, and the rainwater becomes the mosquito breeding grounds, especially to the flies like Aedes, which brings dengue, chikungunya, and Zika virus. So see, we are the one creators for that kind of environment for those mosquitoes to go and breed in the mangroves. because not all mosquitoes are able to breed in the mangroves. So such kind of perceptions, it is time for us to remove such kind of negative perceptions with the help of the school children or with the help of our students, and this message should be communicated to the local people. No, please don't leave this kind of stuff into the mangroves, which is bringing harm to us. We are getting dengue because of the mosquitoes, which are not really supposed to breed in the mangroves. They are breeding because of your plastic there. Such kind of awareness campaigns could bring a good awareness and among the people, local people, and a good importance to and raise the importance on mangrove ecosystem and reduce the destruction to the mangroves. So you all, it is a good opportunity for us. Always the message should be carried to the common people. The science, the message from the science or publication should go to go down to the local community people, because local community people don't have access to read your publications, read your scientific journals. So only the message, they have a mind to grab your message. That's why you have to make some campaigns to create awareness on the mangrove ecosystems, not to release, not to look at them negatively. They have to look at them at positive side of the mangroves, and they must appreciate the goods and services that delivered by the mangrove ecosystem. See here, for example, I saw even sofa set in the mangroves that landed in that is people just discarded sofa and that landed in the mangroves. Not only sofa, I think you can find the refrigerator, television, and whatsoever. So it has become it's like a trapping ground, and please don't do that, for God's sake. And see, sometimes my students make fun out of it, and this guy is writing recording the data when it was suddenly started raining, and then he put a ice box lid as an umbrella and making it, and to get rid of the rainwater to destroy his notebook. So this kind of things are happening, but it is sad that we cannot leave the plastic at any cost. That is the message we really have to give them. And now, when we look into the South Asia, Southeast Asia, and Asia Pacific, unfortunately, Malaysia and also your country, Indonesia, they are the secondary hotspots. That means still we are losing the mangroves. We are losing the mangroves, and also we are in the list of hotspots. So we have to really bring a stop for this kind of things. Mostly they climb to have paddy cultures and palm oil cultures. They are the one mostly affecting our mangrove plantations. So probably we have to think alternatives for to see to reduce the burden on the mangroves. One thing is true, is mangroves is not only the vegetation. Mangrove is a mangrove ecosystem itself is a social ecological system. That means it is not only the plants. There are so many things which you can study, which you can apply the technologies, techniques. So you can study the ecosystem assessment. You can use socioecology. You can look into the species environment relationships, how the crabs are living, which environment is supporting them, and how the ecotourism is benefiting the awareness of mangroves, and how the pollution is impacting the mangroves, how the remote sensing technology and GIS can be useful to identify the mangrove extent or the larger microplastics or marine litter deposit grounds within the mangroves and so forth. So you can plan as a scientist, as a student, we can always plan a novel topics to study your to complete your studies. So it is mangroves is not, don't think always only the vegetation. Vegetation is only a part of the ecosystem. There are so many things that you can explore for your research by using the remote sensing. So as I told you, I am going to say a few observations based on the remote sensing, especially the drone. Sufyan has informed you the benefit of using the space data. Now I tell you the aerial data. Aerial data means that one from the airport, airplanes, or from the drones, how you can make use of them, study the mangroves. And also I wanted to tell you, because I'm sure that being marine scientist, marine scientist and marine scientist students, you all have a great opportunity to visit the mangroves nearby. Maybe you must be sitting next to the mangroves, if I'm correct. So why don't you really use some mobile apps? I'm sure you all have 4G phones and smartphones. Why don't you download some mobile apps and play in the mangroves by using the mobile apps? Just for fun. It is not only fun, give you fun, but also gives you some scientific information how to access them, which were not possible before. Now the different softwares are available as mobile apps for you to make use of them. So as I said, the data which Sufyan has shared with you, it has a problem because of the cloud cover. Sometimes you have a cloud cover, especially to work on the mangrove ecosystems. So, and in those conditions, the airplane, the data obtained from the airplanes or the drones is quite aerial data, is quite useful. Aerial data is also a part of remote sensing. Only thing is you are just using the drones or airplanes with the sensors or cameras to capture the information. So it is a remote sensing, and it is quite useful for you. And the good part with the aerial remote sensing is that you can adjust the height of the flight. So you know to which height. That means you can fly your drone or your flight below the cloud cover, so that you can avoid the cloud cover problem. It is not possible for the satellite data. It is possible for the aerial remote sensing data. So it is a benefit. So many of you must have drones, just plain drones, but you still can make use of them to take the pictures of the habitat, coastal habitats like mangroves. See, this is how a drone picture can show you the different species, mangrove species, availability of different species. For example, when you are in the low tide, when you reach to the island of a mangrove, it is very difficult for you to walk because it is clay loam soil. You might go into the soil up to the waist. So it is difficult for walk for anyone. So at the time, this becomes the drones becomes very easy. You can fly the drone and see what is inside. So now you can see by looking at the different color of the vegetation, you can see different species. And where there is a denudation, that means where the mangroves were cut, or which plant species, or which. So you can, by observing that the tonality is nothing but the color of the image. So by looking at the tonality, it is the terminology that you often use in remote sensing. So by looking at the tonality, you should be able to identify different objects and different species. That is a beauty of the drones also. So here you can see the technology that useful for you to analyze the remote sensing data, the terminology like tonality, texture, structure, shape, size, shade. So these are all the terminologies you use it in the remote sensing. Those are all things also you can detect from the drone images. So this is very interesting that once you are interested in remote sensing, you can use drone data or satellite data for the analysis of remote sensing. So for particular this talk, I just wanted to share very few observations of a drone acquired image on the mangroves. So here, this is the satellite data. You can see PDAS, which is about 5 meters resolution and so forth. So then this is the area which we wanted to test the applicability of the remote sensing data. That means both satellite data and also the drone data. See how clear the drone can show you the image and the objects on the ground. So as I told you, the satellite data, by looking at the PLIDAS, we were able to find only 6 land use land cover classes, which you can see in the blue tone. But when I use the drone data, you can see nearly 10 classes. That means how clear the object on the ground, because the drone data can give you very high resolution picture, which is up to 2 to 3 centimeters. 2 to 3 centimeters. So this image was taken when we fly the drone around 80 to 100 meters height altitude. So at 100 meters, 80 to 100 meters, you still can get 2 to 3 centimeters of resolution. How clear you can see each and every object on the ground. That's why you have a better accuracy, especially for the classification. So mostly when you use, like Sufyan showed you how he classified the phytoplankton, chlorophyll A in the coastal waters, you can also classify the mangrove vegetation based on the satellite data or based on the drone data. Here, I can show you both object-based and pixel-based classification approaches that can give you different results. No doubt, the drone image has given an excellent overall accuracy when compared with the satellite data because of low resolution than the drone image. So again, between object-based and pixel-based classification approaches, the pixel-based classification gave us a better accuracy. So you can go up to more than 90% accuracy for classifying the mangroves, species level classification in the mangroves by using the drone images. Not only that, you can also identify the height of the trees. You can also identify the height of the trees through canopy height model. You can apply a model called canopy height, and you can observe the height of the trees. So in that case, you don't have to survey the whole forest by using the clinometer or hygrometer or laser distometer. So you don't have to use it. So you can make some representative samples for the height examination, and then you can use satellite data or drone data for your height observations. For example, we have did the same thing. We flown the drone at 2 areas in the mangroves. One area is a managed forest. That means the forest department has a plantation, and another area is unmanaged forest. That means forest department didn't do anything. That means it is the natural growth of the forest. Then when we apply the canopy height model for these both areas, the managed forest has a very close correlation with the ground truth values when compared with the unmanaged forest. It is mostly due to the homogeneity and heterogeneity in the forest. That means the forest with a homogeneous cover has ability to give you a better result through the canopy height model. Especially this homogeneous kind of forest, very useful when most of the forests that are subjected to the management. For example, one of the examples you must be knowing, Matang mangrove forest in Malaysia. So Matang mangrove forest is one of the forests that used for from the last 100 years. It is under the management, systematic silvicultural management for the production of the poles, mangrove poles, and also the production of the charcoal. So they make charcoal and poles from the mangroves. And indeed, you must be knowing that Bintuni Bay in Indonesia, Bintuni Bay, also have the mangrove chip production based on 25 years rotation. But in Malaysia, they do 30 years rotation. They do 30 years rotation. So in particularly that kind of forest, the drones are extremely useful. You can one day you can see if there are any illegal activities are going on inside. That means indiscriminate felling of the trees going on, or if what is the height of the trees, and importantly, and also what is the biomass, what could be the biomass of the trees. It is also useful. You can derive the biomass by using the allometric equation with the help of the drone based measurements, canopy height measurements. So again, as I said, the biomass values that derive from the managed forest has given a very close correlated values with the estimates, ground-based estimates when compared with the unmanaged forest. So this drone-based estimates the extraction of the biophysical properties like height and biomass quite useful when the forest is homogeneous, especially the forest which is subject to the silvicultural management. So that is the point which you have a lot of areas must be in Indonesia. So try to make use of the drones for your research. Try to make use of the drones for your scientific activities. So just make it an hobby. Indeed, the mangroves are fueling the livelihoods, right? You know, there are so many people depend on mangroves, especially for the for their daily livelihood, their daily income and livelihood. So this is also a part in Matang mangrove management. There are so many people depend on. So, but what I wanted to highlight in this slide is must be so many social ecological activities going on in Indonesia. Please try to make them, try to analyze them and put them in some sort of a economic flow, cash flow, how much cash is running through that particular activity. In this paper, I analyzed the cash flow techniques for the charcoal and coal industries. But in your area, maybe you see how much social ecological activities are going on in the mangroves and what is the social, how they are extracted for the social, different social activity. So then commercial activity, then try to apply some kind of cash flow dynamics and see the cash flow dynamics, how the people are benefiting, who are benefiting, and what are the regulations to be made, and are there any limitations for that kind of activities. So such kind of things you can still play around to bring up new scientific knowledge to the ongoing management. Yeah, this is the slide which I showed you. The next slide, I'm going to show you some mobile apps which are quite useful for you to use it in the mangrove research. So then you don't need to look for any plastic in the mangroves or any umbrella for you to be away from the rainwater and not to destroy your log notebooks. And most importantly, I want to show you the species identification apps. When you go to Google Play Store, these all apps are available for free downloads, except the app from the Mangrove Watch. I think this app is asking for $5 for the license, but afterwards it is endless, infinite. You can use it for infinite times. But the other apps like French Institute of Pondicherry or Godrej, they're entirely free, and you can install in your smartphone, and you can use them for the identification of different mangrove species. That means you don't have to carry any taxonomic books into the mangroves. Similarly, there is one more software, one more app available that is named after ODK, that is Open Data Kit, that was developed by one of my partners from Aberystwyth University in UK, Professor Richard, and this is available under the Earth track menu. So when you go to this track, you should be able to download this ODK app, and you can really feed your real-time data. When you are really in the field, you don't have to use pen. You can use the buttons, the soft touch screen to feed your data into the system. So during the feed of your data, you don't even need internet. I know mangrove areas don't have good internet connections, but once you download the blank form, you don't need any internet data. You just fill in the data and then you can submit once the internet is available after you come back. So that submission will go to Earth Explorer website, and many people, with your kind permission. If you don't permit, no one can see the data. If you allow them to see any other partners, so your research partners in Australia or America, they all can see the data which you are collecting on the ground in Indonesia or in Malaysia. That means your international linkages, your international scientific collaboration among the students as well as the researchers is going to enhance by using such kind of mobile apps. Just think of it. Think of it. It is very good for your future. Similarly, there is one more app known as GLAMA, that is gap light analysis. Gap light analysis. Earlier, when I was doing my PhD, I used to see, to the canopy and just estimate visually how much, maybe 17% light, 18% light, or 20%. If you have a more canopy gaps, your gamma will be high. So that light penetration into the mangroves, or for that matter to any forest, is very important, because some plants can live shade tolerant. Not all plants can live shade tolerant. The same applies to the animals. Many animals don't like shading environment. Some animals like shading. Some animals want to expose themselves to the sun. So the sunlight is an important, and also sunlight brings the cycle, nutrient cycling. So these all changes the atmosphere in the mangroves, and facilitate the growth of animals, plants, and nutrient cycling, and so forth. So this is all very important, which you can determine just by an app, how much the gap of the light is penetrating into that particular place through the glamour. So you can make use of that kind of mobile app as well. So we have tested this glamour and also the conventional technique. We found very close relation between the values which we observed out of the glamour and also Kajana's cube. That means a traditional technique, how you could measure the gap light analysis. Now we are trying to, my colleague, my research partner, as a proposal that track and glamour are going to fit with each other. That means whatever you measure glamour, that can be incorporated into track as well. That means your all data will be saved at one platform. And now you also, one way I told you, you can use the drone to measure the height. If you don't have a drone, you can also download a mobile app to measure the height of the trees. It is from the NASA, which is the most reliable. You can use that app if you don't have any instrument like clinometer or hygrometer or distometer. You can make use of the height measuring app to measure the tree height. So there are so many advancements are available, and it is our opportunity, is a great opportunity for all of us to make use of them, promote, to encourage, and to encourage the students to make use of such kind of advanced technologies to have an efficient mangrove research. In addition to that, you please think of edutourism, ecotourism, or field schools in the mangroves to promote the mangrove conservation and management wherever is possible. This is the one I'm telling you, the Tropimundo program. Every year we get a lot of Tropimundo students to Malaysia, and they take part in. Indeed, we have a very good student from Indonesia also. She studied with us in Malaysia as part of the Tropimundo program. And that's how we are promoting research on mangroves and conservation. And we have a very strong tie with the local forest department. Then we will bring up new activities to promote the mangrove conservation and management. And also make sure that your students are part of the plantation programs so that they will know. So usually once you plant some trees, how much love you develop towards that particular tree, those who loves gardening, so that they can explain very well. So once you plant the tree, you never think of chopping down a tree. So that's why how we change the mindset. So ask them to promote more plantations so that they don't really feel like cutting the tree in the future. Also promote your activities through community-based programs. You introduce the students to the local communities and raise the awareness of the mangroves, and so that mangroves will be on the safe side. Especially, it is very much needed for Asian countries, because we are still facing a greater part of the mangrove decline in the global estate. And also, I would encourage Indonesia, Malaysia, India, and so forth. We all have superstars. So many superstars and celebrities are there. We can make them. Why don't we make them as ambassadors for the conservation of the mangroves? So I think that is a very good step, so that the message can be reached to the local community people and create a better awareness for the conservation. So finally, as part of MARU, I would like to say that let us protect the mangrove treasure through improved national and international engagements and collaborations. And thank you very much. And if anybody interested to interact with me, you can write an email to me, or you can share your questions through the platform. Thank you. Thank you so much.
Thank you very much. Very interesting presentation from Professor Satya. It's all about mangroves. So if we talk about mangroves, it's not only plant, yeah, not only tree, but also there is a social, economic, yeah, and then livelihood, yeah, from mangrove activity. So, this is Professor Satya share about some application, yeah, that can be used for the mangrove research. So and I think it can improve, yeah, our data, yeah, and information, yeah, related to mangrove condition.
Yeah.
And now it's time for the discussion. I invite students for 1 or 2 or 3 students for questions. Okay, it's already one student raise hand.
Yes.
Please, Irgi Fadilah.
Yes, Fadila.
Okay, thank you very much for
Yes, Fadila.
Mr. Begin for the time. So, good, maybe good morning or good afternoon. Yeah.
Good afternoon.
So my name is Rifadil Rahman, and I'm from Marine Science and Technology in IPB University. I wanted to ask a question. So, in preserving the mangrove ecosystem, I think the role of local resident is the biggest factor. Yeah. But sometimes, in some case, maybe the local people will be interested in preserving it if there is an economic benefit. Maybe as an example, we can make something from its fruit, like pedadas fruit. In Indonesia, we call like buah pedada. It's pedada fruit. I've seen my friend in his competition turning it into syrup or into chips. But my question is, if one day mangrove will start to be recognized from an economic point of view, will this have the potential to cause harm to mangrove conservation in the future? Because maybe this will have the potential to be overexploited. And maybe for the second question, may I, Mr. Begin? Okay. Maybe for the.
So maybe can I explain to your first question if possible,
Oh,
so that
okay. Okay.
you can give me the second question later. Yeah, a very good question you asked me, Mr. Fadila. It is indeed one of the worrying factor for all mangrove country people, for all the mangrove managers. What if the exploitation limits exceeds? For example, I give you a small example. Now we are using the mangroves for black charcoal, right? We make black charcoal. That is what we call it as gold, mangrove gold, that is exported to Japan. But do you know there is a white charcoal? White charcoal, not black charcoal, white charcoal. And Indonesia is also making white charcoal. And Malaysia, we don't have permission to make charcoal, but maybe some illegal activities going on, I don't know. Why the government don't agree to make white charcoal is the production of white charcoal is quite easy. That means, for example, to make a black charcoal out of 20 tons of green wood, you might require nearly out of 40 tons green wood, you might require at least 1 month or one and a half month to make black charcoal. But to convert this same amount of green wood into white charcoal, you can finish it in 15 days. That means with your high production, speed of production of charcoal, you are indirectly increasing the pressure on the forest. That's one of the reasons the government did not allow the white charcoal practice in Malaysia. They are still dependent on black charcoal. And most importantly, definitely, if once our priorities are relaxed, then definitely there will be a harm to the mangroves. So we must be very strict on that kind of conditions, because the charcoal production is not new thing in Malaysia. It is coming up from the last 100, 115, 20 years now. So to change the mindset of the local people who depend on the mangroves from last 100 and 120 years, it is not an easy task. So only thing is we have to optimize the usage of the mangrove. We have to optimize, we have to draw a line where we need to put a stop for the mangrove exploitation. That can brings in, that at least that can allows the things to go on without further degradation. Now we are examining, for example, the carbon, how much the carbon is retained, how much the carbon is released because of this clear felling technique, 30 years rotation. So definitely when you cut the tree, there will be a release of carbon. When you plant the tree, there will be a carbon sequestration. So is it balancing? It is neutral? Are we at the gain or are we at the loss? So such kind of techniques are there to analyze, continuously question our practices and how best we can protect the mangroves. Especially, one thing is true, sir. Without the local people support, we cannot do anything. So the local community are the major players. We really have to show them some sort of a benefit to have a regulated exploitation of the mangroves, or at least a complete protection of the mangroves. Without such benefit, for example, I tell you an example from India. To protect the mangroves, because the fishermen always going to the mangroves to cut the wood for their fuel wood to cook. But at the time, there are some eco-development committees. There are organized, government has developed some eco-development committees, and they try to give some subsidized gas instead of using the fuel wood, to give them some subsidized gas to replace the fuel wood and so forth. So such kind of measures we have to really, every day we have to really think of and implement the measures, not to increase the burden of anthropogenic pressures on the mangrove covers. It is a challenging. It is always challenging. For example, the nearby area people, if they found the palm oil cultivation is more beneficial, so they started encroaching into the mangrove areas, because we also have state lands and federal lands. So state lands, they are occupied for some personal uses. So such kind of things we have to put we have to draw a line to bring them down, at least, if not prevented at all. That's my personal view about reducing the pressure on the mangroves. Yeah, let's go with the second question.
Okay, thank you very much for the answer of the first question. It's really very understandable for me. And maybe for the second question, maybe some of the answer maybe was answering the second question, but I just wanna ask for maybe it can also be an inspiration for us for conserving mangrove. About some programs in Malaysia. What kind of programs are being carried out by the government in the Malaysia, in net Malaysia, and in an effort to maintain sustainability of the mangrove, and
Yeah.
about how to respond to various problems of mangrove loss like that maybe.
Yeah, exactly. Thank you. Thank you. It's also a wonderful question. The forestry department side, mostly they encourage the plantations by involving the local communities and also institutes like universities and NGOs. They're part of it. So that is the normal routine activity. But in addition to that, some knowledge sharing workshops. So, for example, if I have a project, scientific projects supported by local or international funding, so we try to make use of some small budget allotted to conduct a workshop with the local collaborators or local stakeholders. That is very much needed. That means your scientific findings always should be shared with the local communities to have a better impact. That's what we can call it as a citizen science. You may think about the citizen science to enhance the conservation and management directives. Yeah, citizen science is the best. That means you are making the local person as a scientist. That is very much crucial.
Thank you, FG. This is one question related to from Farrell related to mangrove application. So Farrell question is what do we get by buying the mangrove license? Is the
Sorry, sir. Mangrove?
what do we get by buying the mangrove application license?
License. Okay. Okay.
Yeah. And is the available mangrove data upgraded regularly in that application?
Yes.
Farrell curious about have mangrove planting activities in Malaysia involve local communities or not? If so, are there any obstacle in planting mangrove with the locals community? That's the two questions.
Very good. Yeah, yeah. The first question, license, you don't have to buy license for all apps. Nothing. Only one app I showed you for the Mangrove Watch. Indeed, when you type Mangrove Watch in the Google, you can access the data, how much area from different countries, all things are available. But that particular app is available for you to identify the species, different mangrove species, and it is regularly updated. And most importantly, as you all know, mangroves there are not so many species, just 70 to 80 species. So it is almost updated and it is maintained very well if you buy the licensed version. Because it is developed in Australia, so they ask for $5 license. But in that place, you can use for some free mobile apps, no problem. They are also updated. They are also up to date. They are also up to date, and then it should give you what kind of species. You don't have to carry the identification manual into the forest. You can just take your smartphone and identify the species, at least the closest genus that is possible. That first question. The second question, yes, local communities are always informed and part of the plantation program. For example, in the areas like Matang, where the mangroves are cut on a 30 years rotation based, indeed, the local communities are the contractors for the plantations. So the local communities are given a contract to plant the mangroves and to raise the mangroves. But indeed, in other areas where there is no commercial exploitation of the mangroves, are also the local communities are well aware and they are really part of the conservation programs. That is the beauty. And indeed, I would say Malaysia is very one or two sites that has mangrove exploiting for commercial purpose, but rest of the sites are are preserved. There are positive and negative things are also there, but there are norms. They're preserved. They're really protected.
Okay, Farrell, I think it's already answered the questions. Yeah. So from the application, we can get the data and we can use the
Yeah,
data. And then
exactly. And it is free. Most of the applications are free of charge. No
okay.
need to. Only just one. I told you, only one. Identification app.
Yeah. In Indonesia also we have
Could
Monmec,
be excellent.
Monmec mangrove. Yeah. They made by a researcher from Oceanography Institute of Indonesia.
Yeah, I see.
So probably we can share also, yeah, about the
Perfect.
application.
That is the one. Encourage the students, sir. And importantly, the online data recording without taking the book and pen. Just record the
Yes.
data on the OKD, no open data kit, and then submit. And no one can access. I'm promising. No one can access unless you give a access permission. So that
Okay.
is the beauty. So your integrity is no misuse of data anywhere. But that helps you to develop the international contacts. So Indonesia data, if somebody wants to look at it, then you can come up with a joint project together, scientific project, and the students can go exchange, mobility. So many things are the fruits which you can develop later on.
Okay. I think I have a last question for you.
Sir.
I'm just curious, yeah. Where is the most beautiful mangrove in Malaysia that you have visited?
Actually, to my eyes, all mangroves are beautiful, to be frank.
Oh.
All trees are beautiful. Actually, I'm sitting just 200 meters away from mangrove forest here.
Okay.
Just is that. So I use different mangrove forest patches in the country for different purposes. For the teaching purpose, for the students whoever just arrived UMT, I will take them to the campus visit, to the mangroves in the campus. I teach them some basic conditions and characteristics and identification taxonomy. But to explain them how an ecotourism or edutourism can help the mangroves, I take them to Matang. And I take them to Matang again to see how the coal and charcoal productions are made out of mangrove. This is very new for them. They don't know. They never expect how the poles are, because most of them, my students are mostly from the west, so they never see even mangroves also. So that's really curious. So I choose the locations based on the topic of my talk and lesson. To me,
Okay.
all are beautiful. Sir. Mangroves are beautiful.
Yes. Same here, yeah.
Yeah.
In Indonesia, in IPB University, yeah.
Uh-huh.
All the students, yeah, have experience, especially for the marine science student. So we went we go to Kepulauan Seribu, in front of Jakarta Bay for the mangroves. But the mangroves there is yeah, for small islands mangroves, yeah, not
Small
quite
islands, scattered.
scattered, yeah.
Have you been to Bintuni Bay, sir?
Yeah, Bintuni Bay.
Yeah. That is also wonderful place for showing the students the extraction of mangroves for wood chips.
Yes.
Yeah. How commercially they are exploited. So
Yeah.
probably that's a wonderful. Yeah.
Yeah, we hope.
Actually, mangroves is a living laboratory, sir. It is a living
Yeah.
laboratory. Every day we can learn something new. That's why I always say, no, I have a lot more to learn rather than I would say I'm an expert. No, I cannot call myself as an expert. It's still learning. There is no limit and end for it.
Yes.
Yeah.
Okay. I hope.
Thank you.
Thank you very much, Professor Setia,
Thank you, sir.
for the very fruitful lecture today. I hope can inspire students, yeah, who will conduct research or activity with mangrove. And annually we, not annually, yeah, annually we have a program for planting mangrove, in several parts of Indonesia.
Yeah.
So probably student we can share, with you.
Yeah,
and
great,
what
wonderful.
can we improve, for that activity. So student can invite you,
Yeah.
again for discuss a special for a mangrove activity. I think we have chance for you can give insight, for the student activity related to mangrove, and we can improve our activity.
That's good. That's wonderful. Wonderful.
Okay.
Yeah.
Okay, thank you very much.
Thank you, sir. Thanks for
Dr. Professor Satya. Please give love emoticon, for this
terima
lecture.
kasih. Terima kasih.
Okay, terima kasih. On behalf organizer, we say thank you for Professor Satya for this general lecture today. And after this, I think our session is already finished, and I give the next activity to the organizer. Once again, thank you very much.
Thank you so much. Thank you, everyone. All the best. God bless you.
All the best.
Okay, thank you, Bang Begin, and thank you all for coming to this wonderful general lecture. Maybe next time we'll do more like this. I hope you enjoy this, students, all of students, and I hope this general lecture enlighten your day or your thinking process about the remote sensing and also the mangrove. Okay, I think that will be all. Thank you for coming. We will meet you next time. See you. Good afternoon.
Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Kampus terbaik, inspirasi, tebarkan harapan. Kampus terbaik, inovasi, wujudkan impian. Kampus terbaik, integritas, kuatkan tujuan. Kampus terbaik, beri terbaik untuk negeri, untuk dunia, umat manusia dan alam semesta. Mari bersama hadirkan masa depan dalam langkah dan pikiran agar selalu terdepan, berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah, oh majulah. Terus berpacu dalam keunggulan, lincah pembelajar untuk kehidupan. Inilah semangat baru, tekad raih kesempatan, wujudkan kampus terbaik selamanya.
Masa depan dalam langkah dan pikiran agar selalu terdapat berbekal ilmu dan jiwa pengabdian. Deras arus zaman takkan tertahan, majulah melangkah.
Mari bersama